Okay, so I would suggest we're now 100 people. So let's get started. So welcome everyone to our Cambridge Cheminformatics meeting today. So for those of you who didn't join before, it's usually, it's a meeting we actually had for 10 years or so in Cambridge, and usually it was 40 or 50 people from the local biotech companies, universities, research institutes, and so on. And it was usually 40 or 50 people. And today it's maybe 250 or 260 people are registered from all over the world. So it's not to appreciate what COVID brought, of course, but it shows us that we can actually connect in a much different way using, in this case, Zoom. Well, I mean, we were forced to do so, but there's actually benefit in doing so as well, because people can join who couldn't join otherwise. So this is today's meeting. We will have two talks to alert you of the next meeting already. So that will be on the 25th of November. And you can go to this website, c-inf.net. And also I'm circulating a Cheminformatics newsletter, Cambridge Cheminformatics newsletter, which you're very welcome to subscribe to. So there will be events in our area as well as vacancies and so on. And you can go to this website here, drugdiscovery.net, and that will tell you how to subscribe to that. Basically, just send me an email. And the program today is we will have two talks. So the first one will be on deep generative models for structure-based design. So that will be by Morgan Thomas here at the Center for Molecular Informatics in Cambridge. And the second talk will be expanding connectivity between document structures and bioactivity by Chris Southan from TWA2 Informatics. And one thing I would like to alert everyone to, so this meeting will be recorded. So I would like to alert everyone who wants to ask questions, for example. So that has been agreed with the speakers. And this will be the case in order to make the recording available to people afterwards. Because we had one request also from Japan where the time zone really didn't match because in the middle of the night. So we're going to put the video and audio on the website, so c-inf.net. Check afterwards, after the meeting, for details, where you can also look at it again if you wish to. As for questions today, please type your questions in the chat window, and feel free to do so also along the presentation already. And at the end of the presentation, I will then select some of the questions, as many as we can handle in the time available, and ask the person who asked the question to unmute yourself and then to ask the question in person. So using audio and video, if you wish, to make that a bit more interactive from the personal side. After the meeting, so we usually went to the pub here in Cambridge. That's a bit difficult right now, but feel free to stay online anyway. So I will put people into breakout rooms in a random fashion, maybe 10 people per room or so. So there's a good chance you will meet someone you didn't speak to for quite a long time, or meet some new people as well. So please stay online afterwards as well. And also, if you wish to present, so the meetings are open to anyone to participate, but also to present. So the next meeting is in November. We will have meetings again next year. Please let me know if you wish to do so. So everyone's welcome to present work. It doesn't need to be finished. It can be work in progress as well, simply to share where things stand in the cheminformatics, computational chemistry, and related areas, or data analysis, for example, of documents from any of those areas. Okay, and so without further ado, let's go to the talks and to our first speaker. So our first speaker will be Morgan Thomas from the Center for Molecular Informatics at the University of Cambridge. And he's sponsored by Sosei Heptares. He's doing his PhD right now in the University of Cambridge. And he will speak about towards using deep generative models for structure-based design. So I will stop sharing my screen. And then Morgan, you can share yours.
Yeah, sure.
Okay, I stopped sharing. I think you should be able to share yours now.
Yeah.
Yeah, that looks good. Yeah, the screen is good.
Full screen, and
okay.
There we go, in case you want to point to anything.
Good.
Okay, yeah, thanks, Andreas. So today I'll be sharing some of my progress towards using deep generative models for structure-based design. And throughout this presentation, I'll aim to briefly cover, just to make sure that we're all on the same page, what deep generative models are and what is required of a good deep generative model, as well as some of the methods, so how we implement a structure-guided deep generative model and how we use glide molecular docking to guide de novo molecule generation. And then some of the results we obtained, so how in the case of our case study, dopamine receptor D2, we're able to optimize the docking score. And we compare some of the failure modes that we observed to literature findings, and then assess some of the chemical differences between the structure-guided and ligand-guided de novo molecules, and touch upon some of the structural learnings of the structure-guided deep generative model to satisfy important residue interactions for aminogenic receptors. So first of all, what are deep generative models in the context of de novo molecule generation? Well, like any machine learning model, the process starts with the data. So this can either be the building blocks, i.e. heuristically encoded rules and materials required to build molecules. This could be atoms, bonds, or fragments, etc. Or a virtual library of example molecules, such as Kemball's Inc., or perhaps an internal proprietary database. A deep neural network is then trained either on which actions to take to build a sensible molecule, referred to here as action learning, or learns the underlying principles of a library of example molecules, referred to here as distribution learning. This deep neural network can then be used to sample de novo molecules, which are then subsequently tested using something like calculable properties or predictable properties, which then comprises a reward that's used for further optimization. For example, sequential updating of the neural network weights conditional upon that reward. And then this is then iterated over in an optimization loop. And this is analogous to an in silico design, make, test, analyze cycle without the need for the make stage, of course, because it's in silico. And there are some common deep generative model architectures that are important to understand, including recurrent neural networks, autoencoders, generative adversarial networks, and reinforcement learning, which is also commonly incorporated into the preceding architectures as an optimization algorithm. But how do you define what a good deep generative model is, and hence what are good de novo molecules? Well, there are several basic metrics that can help measure this, such as validity. So if your model generates smile strings, does the smile strings translate to a valid molecule? Uniqueness, so has the molecule been sampled before, which is closely related to diversity. So just because a molecule may be unique doesn't necessarily mean that it's diverse from those previously sampled. And also novelty, so does the molecule generated exist in any of the training data used? But just because a molecule may be novel doesn't necessarily mean that it's similar or distinct to the training data. And this can be really important, for example, in a competitive landscape, where you may have a novel molecule due to a novel R group, but not necessarily a novel scaffold, then that's unlikely to be novel enough to progress in that competitive landscape. And these metrics are covered and defined by a couple of benchmarking platforms called Guacamole and Molecular Sets, or MOSES. However, to evaluate goal-directed deep generative models, there aren't really currently any robust guidelines, and standardized benchmarks that have been proposed, for example, in Guacamole, have been commented on as being too trivial. So currently, the evaluation of such deep generative models is typically down to the discrepancy of publication authors. And this is an extremely fast-moving field. There have been many, many model publications since the earliest ones approximately 4 years ago. But the vast majority only use simple calculable properties or ligand-based predictive methods. And this can actually really restrict application in some common drug discovery objectives. So for example, a first-in-class objective by nature may not have any ligand data or enough ligand data to sufficiently train a ligand-based predictive method. Or for example, in a best-in-class objective where you require novel equity over existing intellectual property, then sometimes by using ligands in the public domain, you may bias de novo molecule generation to already existing intellectual property and really failed to generate those novel enough molecules. Which leads to the overarching aim of this work, which is, can we use structural data to guide de novo molecule generation when available, which could mitigate some of these application limitations. So to implement this, with a proof of concept, you could do this using any of the architectures listed before and several structure-based techniques. However, we chose to use a recurrent neural network in conjunction with reinforcement learning optimization. This type of model performs relatively well in the literature, and it's also relatively simple to implement and to train the neural network. Whilst for a structure-based technique, we chose to use molecular docking, as proteochemometric models and shape overlay techniques typically still require ligand data, which still restricts application in those previously mentioned objectives. Although more robust computational techniques like free energy perturbation are just simply too computationally expensive and intractable on a real-life drug discovery timeline. And as a case study, in this work, I used dopamine receptor D2, which is a commonly evaluated target in published models, due to there being many associated ligands in the public domain for retrospective evaluation. But there's also an X-ray crystal structure that was published in 2018 that can be used for molecular docking. And further case studies are ongoing. However, I currently only talk over this particular target in this work. So in particular, the deep generative model that we implement is called Reinvent and was developed by Marcus Olive Kroner and the team at AstraZeneca, and requires a virtual library such as Zinc or Kemble, and trains a recurrent neural network, named here as the prior network, by a maximum likelihood to learn the underlying probability distribution of SMILES symbols in a given sequence. This prior network is then used to initialize an agent network, which is initially an exact copy of the prior network. And then this agent network can sample de novo molecules, which is subsequently scored by a scoring function. The resulting score is then combined with the prior likelihood to give the augmented likelihood. And this agent network is then updated based on the augmented likelihood. And the prior likelihood is important to ensure that the agent doesn't undergo catastrophic forgetting of the underlying chemical principles that the prior network has learned. However, in this work, what we do is we compare two different scoring functions. So the first is the ligand-based QSUM model that was also co-published in this paper by Marcus Olive Kroner and the team at AstraZeneca, which is a support vector machine that was trained on DRD2 actives and inactives extracted from the ESCAPE database, which is a curation of Chem20 and Pubchem. And this model predicts the probability of being active against the DRD2 receptor. And what we do is we compare this to effectively glide molecular docking, where to make this more efficient, we parallelize each ligand in a batch across a compute cluster. And then for each ligand, we enumerate stereocenters, tautomers, and protonation states using ligprep, and then dock into the DRD2 X-ray crystal structure using flexible ligand and standard precision. And out of these variants, we choose the variant with the best docking score to return to the agent for sequential updating. And if we treat molecular docking here as a classification problem, we can directly compare the performance of these scoring functions, where you can see that we choose a decision threshold here of a docking score of minus 8.5, where anything less than this is predicted as active, and anything higher than this predicted as inactive, which was a compromise between maximizing the precision without completely diminishing the recall, where precision translates to a precision of 0.82, for example, translates to 82% of those predicted actives being real actives retrospectively, which is arguably more important in a drug discovery setting because this minimizes false positives, which can be expensive to synthesize and test in vitro or in vivo assays. But still, if you compare the performance metrics of these two scoring functions, you can see that the support vector machine does expectedly outperform glide molecular docking. However, we theorize that it's actually the applicability domain limitations of ligand-based predictive methods that restrict de novo molecule generation to predefined areas of chemical space and narrow areas of chemical space, whereas the lack of applicability domain in the glide docking will allow a deep generative model to find more novel areas of chemical space. But also, if this performs well, it'll also allow application to those objectives that I mentioned earlier, like first in class and best in class, where ligand data isn't available. But of course, structured data is. So more specifically, we asked the question, can docking score be directly optimized? And how does this compare to a standard QSUM model? So we trained the prior recurrent neural network on canonical non-isomeric SMILES from the Zinc database. And we do this following the Moses standardized curation, with the exception of allowing charged molecules. And we also remove any known DRD2 active molecules from the training dataset to make the task slightly harder. The agent was then trained for 3,000 steps with a batch size of 64, and each batch was scored via the previously defined scoring functions, which was a function of also the prior likelihood, giving the augmented likelihood. And for evaluation, we compare the resulting de novo molecules in question, generated by the SVM agent here and the glide agent here, to several other reference data sets, including molecules sampled from the prior network. So this kind of translates to unoptimized de novo molecules, as well as a subset from the training data, a random subset taken from a different database, so Kemble 26 in this case, as well as known DRD2 active and inactive molecules. So firstly, and most importantly, we can actually see sequential improvement of the docking score distribution seen here on the x-axis over the training steps seen here on the y-axis in this ridge block, where effectively at training step 0, this is the prior network, where each docking score distribution is the distribution for molecules in a particular batch. But to put this in context, if you sample some of the 10,000 molecules from the final trained glide-guided agent and compare this to some of the reference data sets, you can really visualize the enrichment of the glide agent-generated molecules in blue here compared to the unoptimized molecules in purple from the prior, and as well as the training subset in pink. But also, you can compare this distribution to the actual distribution of known active molecules that are active against DRD2. And you can see a greater enrichment in docking score in the glide agent-generated molecules than actual known actives. And interestingly, you can also see that the support vector machine, or the ligand-based guided agent, also optimizes the docking score, although it's not explicitly optimized to do so. So this is likely because the SVM agent learns to generate molecules in a similar chemical space to known actives, thereby inheriting a similar docking score distribution. But actually, if we monitor some of the key metrics during training, so training step is on the x-axis here from 0 to 3000, and some of the metrics, for example, uniqueness, you can see a general downward trend of uniqueness per batch in the ligand guided agent of the support vector machine, indicating that the SVM agent actually starts to resample molecules much earlier. And this is also supported by a similar trend and drop in scaffold diversity throughout training, where scaffold diversity is defined as the internal diversity of the Bemis-Merkel scaffolds. And lastly, you can also see a vastly increased Frechette chemnet distance in the support vector machine agent during training versus glide, where the Frechette chemnet distance is the distance between the penultimate layer of the mean and covariance of chemnet. And a larger Frechette chemnet distance to the training data set can indicate a number of failures, including lack of diversity. So this really exemplifies that actually the ligand-guided agent undergoes relatively increased mode failures compared to the structure-guided model. And these kind of ligand-based failure modes have also been evidenced in the literature. For example, Renz et al. Recently published and compared different deep generative models, including a LSTM recurrent neural network and a genetic algorithm that is learned, that is optimized against a particular scoring function seen here in blue against the JAK2 targets. This is a standard QSAR model. But actually, you have some model controls in green and red here, where the green corresponds to the same scoring function initialized with a different seed. And in red, you see the same scoring function, but trained with a different data split of the same data. So actually, you can see that these deep generative models have a preference to actually only optimize molecules towards those specific model parameters. Another recent publication by Amabellino et al., which published guidelines of using recurrent neural networks, show that in a fine-tuning approach, you also see a quick diminishing uniqueness with training. So with training set size here in the x-axis and training epochs here. And it's actually relatively well known that these deep generative models suffer from a lack of uniqueness and diversity. And there's another recent publication by Blaschke et al. With a subsequent improvement in this reinvent framework, where they add basically memory units to try and address this issue of diversity and uniqueness. And they do a nice analysis here where they measure the number of fingerprint analogs to DRD2 active molecules over training iteration. And you can see quite a difference here with the addition of memory units, increasing the number of unique DRD2 analogs generated, hence resulting in an increased diversity. So in this work, I did a similar analysis where I also looked at the difference between the two different scoring functions. And actually, you can see that although these results aren't directly comparable, because there are model parameter differences, you can actually see that there's a difference in the scoring functions. And it's actually the glide agent that generates more DRD2 analogs compared to the SVM agent, showing that actually you can increase diversity just based on this difference in scoring function. So do these models actually rediscover any explicitly defined known active molecules? Well, actually, they both do, but only at larger sample sizes, and the rate of which is relatively comparable between the structure-guided and the ligand-guided models. But actually, there are no co-generated known DRD2 active molecules, i.e., they both generate active molecules, but they're different active molecules. And there's only one shared scaffold shown here between the two. But actually, if you look deeper at these 10,000 sampled molecules between glide and the support vector machine agents, there are actually no co-generated molecules, which really capitulates the enormity of chemical space here. So then I wondered whether they're at least more similar to known active molecules, where this similarity matrix shows the average nearest neighbor similarity, where an increased number suggests an increased similarity between data sets. And first of all, you can see that it's actually the random data set that's most similar to known actives, which isn't that surprising considering this was taken from Chembl, and DRD2 associated ligands likely comprise a large proportion of Chembl, and there are actually 25 co-occurring molecules here. But interestingly, the glide and support vector machine agent-generated molecules aren't particularly more similar to actives than the prior, although if you look at their similarity to the inactives, they are more dissimilar to known inactives. And aside from this, you can see that glide is most similar to the prior, and glide is most dissimilar to the support vector machine, again, iterating the complementarity between these de novo molecules generated by the two different approaches. And to further try and understand the chemical space occupied by these two different approaches, I show here a uniform manifold approximation and projection calculated using the molecular fingerprints of all the reference data sets. And you can actually see the actives in green, the glide agent generated molecules in blue, the prior unoptimized molecules in pink, and the SVM agent generated molecules in gray. And the glide and SVM agent generated molecules tend to occupy a different region of chemical space. But the glide agent generated molecules tends to have a greater overlap with the prior. And if you annotate the docking score here, as expected, you can see that those areas of chemical space with an increased docking score here correspond mostly to the glide region of chemical space. And to further try and understand the type of chemotypes embedded in this low dimensionality embedding, I display here the centroid of the largest cluster within the annotated regions. So here, if you look at some of the actives across the embedding, you can see that there's similarity between the chemotypes where you usually have sort of capping of the molecule by either a mono or a bicycle. And then these are linked with an aliphatic linker, typically comprising a propyridine or a propyrazine moiety. And then if we look at some of the glide agent generated molecules, you can see some similarities here, where you still get that capping by a mono or bicycle, although there's less focus on the propyridine propyrazine moiety, but there's still maintenance of a charged amine. Then, if we look at some of the support vector machine agent-generated molecules, you can see that they're slightly more distinct. Where you have a similar chemotype, there is a difference in the vector of substitution on any of the aliphatic rings in the linker. Or they're slightly more distinct with a bicycle and then just the aliphatic ring here, and or some kind of combination of the two that you can see here. And to try and further understand actual physicochemical space of the molecules generated, a similar UMAP projection was calculated using RD-kit physicochemical descriptors, where you can actually see that the active molecules and the support vector machine agent-generated molecules occupy relatively similar regions of physicochemical space. But actually the glide-generated molecules and the prior unoptimized molecules show much greater diversity in physicochemical space. And if you look at some of those descriptors that correlate with the embedding, you can see that these two different data sets also generate molecules with a lower ring count, an increased total polar surface area, and a lower molecular weight. And upon clustering of each of these data sets, you can further understand that actually the support vector machine agent generated molecules has the largest cluster size of around 400 molecules, indicating less diversity. And if you focus on the region below that Docker score threshold that we set earlier of -8.5, so that we understand the precision and recall you get with that kind of threshold, and a cluster size above a cluster size of 10, you get enrichment of these virtual chemical series with this structure-guided approach, where there's only 2 or 3 examples of either known actives or the ligand-guided agent-generated molecules. Again, you can see a kind of similar chemotype between the molecules, similar to actives, that is. So actually, how does this relate in terms of structural understanding and the binding pose? First of all, you can see that the bound pose of some of these generated molecules does closely recapitulate the co-crystallized complex of risperidone in DRD2. But you can also see that it learns to maintain this crucial interaction with ASP114, which is a highly conserved residue in aminergic receptors and is really important for ligand binding affinity. And if we further try and understand this by calculating the structure interaction fingerprints involved in all of the bound poses and all these different reference data sets, you can see that the ratio of hydrogen bond acceptor, charged hydrogen bond acceptor interactions made with this aspartic acid residue is much greater in the glide agent generated molecules than any of the others. And something that's not actually captured in this particular technique of fingerprinting is the number of interactions made with this ASP114 residue, where there are actually several molecules that make more than one interaction with this ASP114 residue, which is not typically seen in any of the other data sets. And it'd be interesting to try and experimentally validate this, as if this doesn't translate to activity, it could be a trivial way that the deep generative model has found to optimize docking score. And summarizing these structure interaction fingerprints, you can actually see that in both the glide agent generated molecules and the SVM agent generated molecules, increased interaction with residues deeper in the pocket and decreased interactions with residues shallower in the pocket. And this is likely a confounding effect, just simply based on the training data curation, which had a lower molecular weight filter. But interestingly, you can actually see that as opposed to trying to generate larger molecules to optimize docking score, the glide agent tends to generate molecules with a slightly lower molecular weight, which is quite interesting. As if docking score does translate to activity, which is a big if, then the molecules could have a greater ligand efficiency. So how could this kind of approach be used in real life applications? I think most obviously in hit finding or lead discovery, where, for example, if you have structural data, but you don't have ligand data, or perhaps you don't want to bias your de novo molecule generation towards known ligand data, then you could use this here, where in this example, the training subset of zinc is representative of a virtual screening library that you could then optimize towards a particular target of interest and get vast enrichment in hits above a certain threshold. Or potentially you could also use this in lead optimization by also restricting similarity to a lead compound, but still optimizing the docking score. Or perhaps in knowledge-driven lead discovery by incorporating prior knowledge of particular residue interactions or water interactions that might be important for ligand affinity or ligand selectivity. And building upon that, potentially using different closely related receptors such as adenosine 1 and adenosine 2A, whereby you could dock in both and optimize the docking score difference to try and achieve selective lead discovery. And to conclude this work so far, I think that this shows, in this particular case study anyway, that reinforcement learning can actually be used to directly optimize more complex scoring functions such as docking score. But also that the structure-guided deep generative models can be as fit for purpose as the ligand-guided models and may offer several advantages such as increased diversity. And this also suggests that the binding pocket and glide force field infer enough chemical information to reproduce known actives. And perhaps that actually some of the failures that we see in deep generative models may be more attributable to the periphery scoring function design compared to the models themselves. However, I understand that more work is required, as this is only one case study, and therefore ongoing work is required and is ongoing to more robustly understand the potential benefits we could get from this kind of approach. And lastly, I'd just like to thank the computational chemistry team at Sosei Heptares, including my industrial supervisor, Chris de Graaf, as well as the Bender Group here in Cambridge, and my academic supervisor, Andreas. Thanks for listening, and I'm happy to take any questions, either online or offline.
Great, thanks a lot, Morgan. Yeah, there were a couple of questions. Let's start with the one by Rocio. If you don't mind, maybe you can unmute yourself and ask the question directly.
Hi, Morgan. Thanks for the talk. Can everybody hear me?
Yes.
Yeah.
Okay, good. Yeah, I was wondering, first of all, how many samples did you generate with each of the models for these analyses?
So I generated 10,000 samples.
Awesome. Okay, because then it ties into my question. I think on slide 15, you showed that the GLIDE and SVM models don't necessarily reproduce the same actives, which was interesting. And I was wondering if instead, did you try calculating the likelihood for the actives using both models, since you know what the actives are already, and then comparing what the likelihoods are?
So I've not directly done that analysis, but I must say that actually for the 10,000 sample sizes, neither of the models did reproduce known actives. It was only at larger sample sizes of 100,000 and 1 million samples, molecules sampled from these different models that have reproduced known actives, although I've not calculated the likelihood.
Okay, now it would just be interesting to see, because maybe it was just not enough samples to see the same actives reproduced by both of the models. So it would be interesting to compare what the likelihoods are using each model, and then maybe you can plot them and compare them that way. Just an idea.
Yeah, no, I definitely should do that, actually. Yeah, thank you.
Okay, thank you.
Yeah, thank you for your question, Rocio. There's one by Chris Southan as well. I think he's personally out, but I can read the question from my end. So how would you sample different sets from GLIDE and SVM to then get them made and assayed? Because you have iterations, but quite large sets, right? So I think the question is, how do you handle that in practice to pick which compounds actually to make?
Yeah, so I think that's quite a difficult question to answer, and that's a more general problem with this kind of field. But in particular, something I'd mention with regards to that is actually one of the techniques I think to use to filter molecules for actual synthesis and testing when using some of the traditional ligand-based methods would be to dock molecules anyway. So actually, you're already doing this in this method, but also without prospectively doing it myself on other principles such as SA score, which obviously doesn't always translate to anything meaningful, but also just looking at the same log P region. But I think the physicochemical diversity of this approach shows that you have more choice with the structure-guided model.
Mm. Oh yeah, thanks, Morgan. Chris, I've just seen you were actually there. Your camera was just your face was not in the center of the picture. So
right.
Was that your question, or do you want to have any follow-up questions?
No, that's fine. Clearly, it's screening data is the ultimate test of those the predictions.
Yeah, no, that's key. And we can, I think, do one or two more questions. Abik Seal was asking a question regarding the use of pharmacophoric features. Abik, do you want to ask your question?
Thanks, Andres. So, did you guys test about what are the pharmacophoric features which get sampled from this agent, like the orientation of this 3D pharmacophores for donors, acceptors, and also the aromatic rings, and also uh, how are they different from Glide and SVM?
So I've not actually done that analysis myself yet, although that would be an interesting analysis to do.
Because that is a 3D and this is SVM, I believe, is taking 2D, constraints. So
exactly,
it will be interesting to see.
It would be, yeah. So far, I've only looked at the protein ligand interaction fingerprints, but you can only gain limited knowledge from that. So yeah, I could do that kind of pharmacophoric analysis to try and understand the SAR around at least optimizing docking score, which is something that I guess wasn't typically done before degenerative models. So we could at least understand, what features are needed to really optimize that docking score. And that may not necessarily translate to activity, which is the ultimate question. But yeah, no, I should do that analysis. That'd be interesting.
Mm-hmm. Yeah, thank you for your question and the answer. Let's do one more question, and then I think we move on to the next talk. So there's a question by Abdul Karim Najar regarding exclusion criteria. Abdul Karim, would you mind asking a question?
Yes, thanks, Andreas, and thanks, Morgan, for this nice talk. I'm just wondering, have you used any criteria to exclude, what kind of the chemical space you are focusing during the generation of the molecule in both sides for Glide and in also as well as with SVM?
So
is it molecular weight or what kind of exclusion criteria?
So currently there's no exclusion criteria, as this is kind of a proof of concept study, just fiercely comparing the differences between the two different approaches.
Do you think if we have used one or more exclusion criteria could have a better prediction, a better somehow retrieving of the actives? Or
yes,
similar?
Potentially, yeah, because in reality, whereby, if you build a ligand-based predictive model, those ligands are typically biased by our criteria that we apply on them anyway. So the ligand-based model will implicitly contain that kind of criteria, whereas the Glide model doesn't necessarily. So I believe that if you added some of that criteria again to the Glide model to make it explicit, for example, a particular range of log P, you might actually get more similarity to those known actives. Although that's not something that I've done in this work.
Yeah, and interestingly, because you mentioned, Abdul Karim, you mentioned molecular weight cutoffs. Morgan, what you did not observe, it was not growth, right? So it was not lipophilicity driven. You did not observe that, right? Interestingly.
Exactly, yeah. So actually, they were slightly smaller molecules, which was against my intuition, and also increased total polar surface area, which again, I'd expect increased log P, yeah.
Yeah. Okay, very good. Thanks a lot. Excellent, Morgan. Thank you for your talk and for the questions and the discussion. Thanks a lot. So what I would suggest is let's move on to the second talk today then, and that's by Chris Southan. Chris, would you mind sharing your screen?
Okay, let's see. Ooh. Aha, good. Okay. So we all set?
Yeah, just give it 1 second, but it says it's you start screen sharing. Just takes a few seconds, I think.
Yeah, I got the rolling circle.
Yeah, so far it's not coming. Did you share the desktop or the application? Or?
Screen.
But maybe stop and share the desktop. Usually that works better. Sometimes the application doesn't work. Yeah, now it looks good. Yeah, if you
Okay,
start
right.
the slideshow, it should work.
Yeah,
Great.
okay.
Thank you.
Put that down. Start slideshow. Just adjust my camera slightly. Good. Right, so I'm all live. Yeah, thanks to Andreas for the invitation. For the first time, I put my slides up to Zendo, since I suppose some of us are bailing out of SlideShare. Now it's gone to another owner. You can find out about me on LinkedIn, of course. I've spent 5 years as senior cheminformatician for the Guide to Pharmacology. I'm glad to say I've actually come back for a shorter term position for COVID-19 curation, financed by the Wellcome Trust. TW2 Informatics is my consultancy. So let's go.
I think, Chris, maybe start the slideshow so it shows full screen, because right now it's
okay. What do we got?
Now if you go to slideshow from beginning, that yeah, good.
There we go. Right. Okay. The chemistry to biology join. Let's so I can go up. Yeah. This is intuitively obvious to all of us. We all read papers, work out what the assay design was, the result table, what the chemical structures were, and what the target protein was that this publication was about. So we're after, or we want to capture any kind of chemistry with significant bioactivity in vitro, in cells, in vivo, in clinico. Doesn't have to be good for us. Poisons and toxins are also interesting bioactivity. And this is what we look at for drug discovery, anti-infectives, system stuff, and metabolomics, and lots of other things. So I think that DARCP, document assay result compound approach, is a useful shorthand for the molecular mechanism of action. It clearly has its limitations. It doesn't encompass phenotypic assays, but you can put something else instead of P, or indirect mechanism of actions. Heparin binds to antithrombin 3 and then inhibits thrombin. Covalent inhibition, and an interesting problem of activators of increased enzyme activity, which you want to do for gain of function. So we've got a lost connectivity problem. I was trying to attribute this quotation, which I thought was from Amos Byrok, but he doesn't actually claim it, so we don't know where it comes from. But something along the lines of it's pretty obvious. We spent millions putting data into PDFs, and now we're spending millions more trying to take it back out. So we have 50 or whatever, many years of this legacy. It's intriguing to guesstimate what the upper limits of the quality cuts of the capture that we want to go for. It's maybe in the order of 250,000 papers, maybe 100,000 patents, and I'll show you some commercial figures later on, but somewhere between 5 and 8 million bioactive molecular structures, and mapped to the order of 4,000 human proteins and about 2,000 at least for other species. So historically, most quantitative data is entombed in papers and patents, and only a small proportion is openly curated. This is just a fun slide. We all know the analogy between the PDF and the hamburger, that you put lock stuff in. So this is running the gauntlet to extract the structures the SIR and the database IDs, and if you can resolve them and link them all up. Okay, for my sins, I'm actually trying for the election for an ISB committee member again this autumn, but this is about artisanal expert extraction by biocurators. Now, clearly, if it's a nice paper written clearly, all the entities very clear, on a good day, the biocurator can extract the DARCP in minutes. If it's a fairly gnarly patent, it can take an hour or so. But we often encounter accidental obfuscation, all types of errors by journals, anything you can possibly imagine. Orders, 3 orders of magnitude wrong in units, etc., wrong gene names. And of course, for a proportion of patent applicants, they will deliberately obfuscate the relationships inside documents, which make it hard work, but they have to declare the reduction to practice. So activity results will vary within orders of magnitude between publications ostensibly for the same compounds. Now, it's very difficult to judge the quality and the potential reproducibility of any document that you want to select for curation, and this quality is a big issue. I.e., we probably don't want to capture everything, and there's variable publisher guidelines or for authors to disambiguate their entities when they're in the paper. So many chemical structures from nature to science, everything else, are just images, 2D images in the PDFs. And results are sometimes buried in supplementary data. They become data tombs. Limited author awareness of ontologies and protein naming. And last but not least, of course, poor sustainability of the funding and career structures. So I'm going to review the what I think are the 4 main DARCP curated open databases that all 4 of them submit to Pubchem. Guide to Pharmacology, won't go through all the figures. This is the number of publications that the guide points to. Some of them are not. This is the core set, about 17,000, wherein the binding constants are specified in those papers. So we've got a total of about just over 1,500 human targets where we have quantitative interactions between the ligands. Some other figures here. Might be slightly surprising that the primary targets of approved drugs are quite low, under 350. But if you look at the secondary interactions as well, you go up. So just while we're on the guide to pharmacology, this is in a sense the linking, reciprocal linking between Pubchem, PubMed, and the guide to pharmacology. Now, as I mentioned in our chat and prelude, we had a nasty shock about 6 months ago, about 3 months ago, when these links, these entree links that were they're in the right-hand facet of PubMed. You could go straight to these links. Suddenly, they disappeared. Lots of backstory to this, mainly that the new improved PubMed was built out on a cloud instance, which is very different, separate from the entree system. Anyway, I'm pleased to see they've actually returned in a slightly muted form right at the bottom of the record. But you can now, so from a PubMed ID, you can go to Pubchem compound or Pubchem substance. And in this case, it happens to be a coronavirus, very recent coronavirus paper. And these 2 compounds were curated by my colleague, Elena Facenda. So we have the biological activity in the guide to pharmacology entry. So these sets are reciprocal. You can come in at any entry point. You can come in via Pubchem. You can come in via the guide to pharmacology, or you can come in via PubMed. Chembl, most of us probably very familiar with Chembl, fantastic achievement at scale. This is their overall stats, but it's important to note that actually a rather smaller set are actually curated from the literature, and they import. I think in retrospect, this is not looking, doesn't look. This has issues associated with circularity of submissions and coming of the resubmissions of assays are already there. But anyway, about 500,000 compounds from Pubchem bioassays. Targets, this is not just proteins, this is cells and microorganisms and stuff. 76,000 documents. So I'll come back to this later. Binding database, these are their statistics for their release, about 0.8 million. They have BindingDB and Chembl have reciprocal mirroring arrangements. So this BindingDB takes some of the stringently protein mapped entries from Chembl. Chembl takes the patents that are curated by BindingDB. So BindingDB is unique in as much as they curate Dlcp from patents to get the activity. This is the stats from this month I got from Michael Gilson. So a substantial chunk of patents, quite significant. So it's 0.3 million, and these all have they all have SAR. That was why they were selected. The example numbers, i.e. a location within the patent document, they come through to Pubchem. It includes cross-screening targets, i.e. you see a set of HGNC numbers for all the assays that were included in that patent document. So 7 compounds multiplexed out to 47 sets of affinity data. Here's an older example, inhibitors, so 0.32 nanomolar. So this is the level of resolution you get, and these are all mapped into Pubchem. Data is mirrored in the Chembl releases, but they've got a longer release time. So we can look at the comparative statistics of open Dlcp. This is from the publication, which I'll show you at the end. So on simply the publication level, this was actually back in January, so it's a little bit out of date. I had to get the Chembl, I got the Chembl cross references from European PubMed Central. So you can see there is a strong difference here. So between these three, you've got unique capture and of course overlap. So they've all three of them have they've curated these papers in common. They won't always get the same compound out of the same paper. Certainly BindingDB, most of them should generally get the same lead. Guide to Pharmacology tends to focus on the lead or 1 or 2 compounds. Whereas Chembl will usually take the whole SAR set. Chemistry. You can do these intersects in Pubchem. Not surprisingly that Chembl has the lion's share of chemistry extractions, but there are unique extractions in each of the sources. We can discuss the regions of uniqueness. Part of this, certainly for BindingDB, comes from their patent extraction. So remember, the ore total for these three comes just up to 2 million of active compounds that are ascribed to targets in these 3 databases. Now, if you look at the proteins, there's a very nice resource that well, I've encouraged us to get into, and I've encouraged some other sources to get the cross-references, but SwissProt will collate the cross-references to protein identifiers from these, in fact, these 1, 2, 3, 4, 6 sources that curate chemistry. Now, Drug Central tends to focus on drug targets. I won't go into any detail with that. Drug Bank Drug bank tends to have a slightly lower stringency of mapping. So these, to an extent, you get a certain amount of over mapping. This is these were the all species selections. Now, Swiss lipids is interesting. I haven't put it in the analysis, but Swiss lipids maps to the metabolites, which are the lipids, and it maps them to the enzymes, to 1,341 enzymes. So and you can slice and dice these to compare, but they do have different mapping stringencies, different mapping rules, stringencies, and update cycles. Although they all update with it's independent of the SwissBot releases every month. Okay, intersecting target. So now I'm using the SwissBot intersects to look at the target differences between these three. So here again, we see uniqueness in each set, different reasons for the uniqueness. I suspect a lot of these from guide to pharmacology are the historical peptide curations that we've been doing against receptors for a long time. And of course, there is consensus and divergence at the same time. And if you add these up, you've got about 18% of the protein, 18% of the uniprot proteome, although that's inflated by at least 2,000, but that's a different matter. Okay, now these three merge into Pubchem. Now, Pubchem is, Pubchem's amazing. It is a de facto global portal, and since the latest patent additions, it's now up to 111 million number of assays. Most of these actually come from Kemble. And the bioactivity. I take the genes and protein numbers with a pinch of salt, essentially. There's an element of overmapping by orthology. Now, this is very high. According to these figures, no less than about 3/4 of the whole of PubMed has some kind of chemistry links. But this is because of common chemistry. So there's a lot of mapping there. Now, patents, 3 million patents. The patent number is very redundant, for those of you who know about patent families. I'll come back to that later. And there are seven lots of data sources contributing, mostly substances, but sometimes annotations as well. Now, just at the end of this section, I'll go to there are a number of commercial sources which will offer you, they'll sell you access to their Dlcp extraction. Probably one of the leading ones of these, because I've actually collaborated with them in the past, is Accelera GoStar. Accelera used to be GVK Bio. So they've come up with about 8 million compounds in their system, extracted from about 80,000 patents and close to 200,000 journal papers. Okay, and their numbers, they come out to 9.3 million assays, 10,000 targets, functional assays, and this is their in vivo to in vitro split. So whilst this is a licensed access to this data, it kind of indicates what the upper limits and what the public sector could extend to. They have, of course, how do they get so many? It's basically curator numbers over the years. So a little bit on not activity mapping or targets, but simply chemistry to document, or what I call C to D. So it's kind of a gateway to the full Monty, but it has some limitations. But it is it's quite important. Key publication back in 2016 about Pubchem association between PubMed and Pubchem mappings. So interesting enough, and not obvious, perhaps a little counterintuitive, the lion's share of these was contributed by the IBM patent extraction effort, which also extracted abstracts in parallel with the patent. So that's why you see these. NID, that's their infectious diseases curation from the US government. This was us. We got a little honorable mention there, and there's some other sources in there. Now, if you go up to today updates, you can see, you can read off. This is the list of substances, and these top four are patents, in fact. So Shaw Kemble cranking them out down in Hingston, 20 million compounds. New one just arrived a few weeks ago is Google Patents, 19 million. PatentScope from WIPO has Infochem extraction technology on the back end, 16 million. IBM, but they ceased in 2017. I doubt if they're going to do a refresh. For various reasons, Springer and team piled in with extracting no less than 0.6 million substances from their papers, and this is their annotations in the Pubchem CID records. Team, actually, the team chemistry folks with their journals and book articles outdid them slightly, so they got even more. So these are very useful, but they have the caveats. It's very difficult to, you don't get SAR, it's not full DARCP, and you have the issue of the document aboutness. Yeah, you've got a compound document mapping, but is that document about this compound or whatever else? And it's getting complicated because you've got indexing not only against the PubMed IDs, but also you've got the DOI, and some journals are a DOI only. And there's a confusing set of parallel compound to document mappings inside Pubchem, particularly, IBM and Mesh. Now, patent extraction now has hit 34 million compound IDs. That's 34 million. That's over 30% of the whole of Pubchem. But there are associated quality issues of automated extraction, and certainly for patents, and to an extent for the documents as well. You've got this problem of massive futile indexing of common chemistry. They just go round and round indexing common compounds. For example, you can find 140,000 patents on aspirin. I defy anyone to try and make sense out of those in an IP perspective. So what's next? The title was advances or progress in the sense, but let's make a bit of a review. Now, FAIR. I haven't got time to explain FAIR. It's a big thing. I suspect most of the audience has already heard of it. Findable, accessible, interoperable, and reusable. So this is a big thing now for funders and publishers and authors. And funnily enough, it's been adapted even inside commercial operations, that they also want to be fair. So this should, in theory, increase the transfer of the entities, documents, assay result, compound, protein, should increase their transfer into repositories, but not necessarily in their linked form. However, I posit there is a labyrinth of deposition options, which even I find confusing. So I don't know what authors are going to choose to what they put where in the pursuit of fairness for their undertakings, including their publications. So there's a potential of FAIR to increase the flow of linked DARCP. That's why I put the hyphens in between, that the linked relationships into major databases such as Pubchem assay, but we'll see. There's no evidence of a flood of this so far. So, how are we doing for time, Andreas?
No, it's fine. I mean, it's just continue. It's fine.
I will. Yeah, I've only got a few more. Right. So, council of perfection. It's an old phrase. It's not council. It's spelled council. Okay. This it's obvious. Okay. So we mandate authors to explicitly connect their own DARCP results in a form that is fair, extrinsic to the PDF, for goodness sake, outside the PDF, structured with the metadata, machine readable, ontologized, and transferable to open database records and reciprocally linked to the publication. Now that's a quote from me. So we all know authors need to become their own biocurators. It's been technically feasible for decades, but even now, not one single journal insists on authors providing a machine-readable DARCLP flow. Sorry, L means location of the structure in the document into, for example, Pubchem bioassay. So yeah, we know what the impediments are, sociological factors, and the historical publishing models. So the bioscience community now, importantly, as there's all the rage now, because we include the AI aficionados. Up to an extent, they still have their feet nailed to the floor because of decades of DARCP value entombed behind firewalls and buried in patents. Now, as we all know, curated resources, particularly the four I've mentioned, they're crucial, but they need more resourcing. Yeah, well, automated extraction, NLP, and all that jazz. Maybe it's coming towards automated extraction, but at the moment it remains way behind the specificity and the quality selection of expert biocurators, and it's the quality that underpins the AI modeling. All right. Now, the existence of parallel document chemistry linkages in Pubchem, as well as these automated lookups of compound names in PubMed Central and European, they are useful, but they can be quite confusing. So the publishers should step up to better facilitate the DARCP capture, in a sense, as part of the fairness campaign. Okay, so I'm finishing up now. Look forward to some questions, a few papers. Some of what I've just described, in fact, you saw some of the diagrams, are in this Bellstein article, which I was pleased to get out in the beginning of this year. This is the latest nucleic acids research database issue from Guide to Pharmacology. Little bit by us in a couple of years ago on some of the challenges of connecting chemistry to pharmacology, same kind of thing. And this was some early work on that commercial database, the Ghost Star, but it's quite old now, but it provides some good background on the way they approach things. And there's a few of my blog posts may be related to this theme and of interest. So thank you very much. I think that's it, Andreas. Great,
yeah. Thanks a lot, Chris. Yeah, chemical and biological data is certainly a tricky topic. And there's a question, or actually two questions from Peter Mararast. Peter, do you have, there's applause as well, I've seen, so well done. Peter, can you unmute yourself? Would you like to ask a question?
Hi, can you hear me okay?
Yes, hello.
Okay, hi, Chris. Thanks very much for a very useful overview. And your last slide, I think, was key. So we can do this. We have the technology to do it. Yeah. We don't have the political will. I don't think we will easily have the political will, but we are gradually collecting enough common technology that we can try and force people's hands on this. And this is what I think we should do. So you asked about which of these things can we link together. Compounds and proteins are well done. They've been done for 30 years. Documents are getting better with people using JATS so that the semantics of the document are well understood.
What's that, Peter? JATS?
It's XML. It used to be called NLM-DTD. It's now called JATS, and it's 250 tags for a scientific document. So,
a tagging thing. Yeah.
So it's actually marked up very well by PubMed Central and distributed in Europe, PubMed Central. So, for example, you can go straight to the addresses of the authors or the name of the reviewer or the fifth reference and get the authors and their locations from that. It's very well done. It's not so well done for the body of the text, so you generally get a rather patchy introduction, methods, results, discussion, and you have to work that out yourself. But your other two, are there any agreed semantics in the community? Because if you don't have those, you can't get people publishing them. So remind us how A and R are supported at the moment.
Assays and results there. There's been, well, for example, Strenda, which is the, if anyone doesn't know the term, it's the guidelines by the Brenda enzymology people. So there are lots of, we even co-authored, what do we, MIAPI, minimum annotation for protein compound interaction stuff. So there are guidelines out there. I mean, empirically Campbell captures this. Okay, so it is in a sense outsourced annotation, but that's what you get back. So you get results. But there are, as you say, there's a shortage of standards for activators and covalent inactivation and stuff like that. So no, there's a further to go with assay descriptions. There are various efforts at assay ontologies, but I can't see a big take up of them yet.
Well, that's a pity because we have to have that if we're going to have any reasonable AI working on this. If you've only got a textual string of activity, it's very, very difficult. And I think the only way it will come is by the appropriate sub-communities creating specifications for, what is a particular receptor test in some rat model or whatever.
Absolutely. But we used to have this, I remember discussions with Steve Bryant in the past, that the problem is that the experimental community are very creative. So whatever assay ontology you come up with today, they'll invent a new assay tomorrow.
Yeah.
Yeah. Okay. Thank you. Thanks, Peter. We have one more comment as well from Karthikeyan. Didn't hear from you for years. Karthikeyan, would you like to make a comment verbally as well?
No. Still muted.
Hello, can you hear me?
Yes.
Yes, hi.
I was very happy to hear both Peter and Chris, and thanks, Andreas, for this fantastic presentation opportunity for all of us to understand how to destroy a document using PDF format. And yeah, that is a right way of saying, actually.
Hamburgerization. I think was that your invention, Peter?
It was credited to me, but it's actually Michael Kay.
Okay. All right.
So today we are all suffering because of these publishers. Luck means their interest on PDF format, and even full text is not helping us. Today AA is required, and today we want antiviral and we want all the molecule. There is no tool available. There is no format available. So we
Yeah,
are all
no,
suffering because of that.
yeah.
So
No,
we are
no
not
smile
able.
strings, no smile strings in the PDFs. On the,
So today,
you might get IUPAC on a good day. Yeah.
yeah. So today we are suffering because of our own, issues. What we made it. So I think based on this comment, I think Peter did a lot of work on CML, and people are talking about XML. So if this HTML, PDF, DOI, everything is linked to molecular, bioactivity, etc., it will be a great resource in at least next 20 years to come. So that is what my comment is. So we have very difficulty in doing text mining. So you can't do a perfect text mining. We are using Oscar 4. We are using LinkPipe. It's all very difficult today to harvest data from the medical literature. So it's a very fantastic, eye-opening document presentation. I really thank you, Chris, for your nice presentation.
Cheers, cheers.
Yeah.
So can I respond to that? Thank you very much, Kathy, for that, and Chris. I'm optimistic that we could get a bottom-up project that at least creates proof of concept for the extraction of chemistry from the current literature, both from names. We now have a lot of material that allows us to build an ontology for chemical names, which when coupled with OPSIN allows us to extract a lot of names from the literature. And also we're doing some recent work on extracting data from graphs, and that would be things like drug response, dose response curves and things of that sort. So if there are people who spend any of their time extracting material from the current literature, I'd be very interested in talking with you, because we are automating our workflow. And at the moment, for example, we're looking at extracting NMR and crystallography from the literature. And that's with a group in Cambridge, which is looking at battery materials. Claire Gray's group, Matthew Dunstan, and we're building tools which will take these diagrams and extract. So if you have standard diagrams, standard tables, then there's a chance that we can get things out of it. It makes a very good project, for example, for an MSc student who's learning chemical informatics, and we can guarantee that you can get something out in a few months' time.
Okay, so very good. So get in touch with Peter in this case, right? That is, yeah, for everyone. Okay, thank you. And there's a comment by Stefan as well, who was commenting on some of the points made earlier. Stefan, would you like to verbalize them?
Well, just because it was sort of suggested that authors and or publishers need to do something about it and basically publish in a better format.
We're still waiting.
And yes, I think great idea, but we've tried this and it's incredibly difficult to get.
I couldn't agree more. Who are you? Who are you, Stefan?
I've been working with Peter, for example, and Asa, I think NMR and NMR databases and so on.
Great.
And it would be great if you would at least have a peak list in a machine readable format alongside your.
But can't, in your case, can't you harangue the instrument manufacturers so you can pipe it out?
Yes, but that would require for the publisher to require authors or authors voluntarily depositing those data, in some sort of standard repository or whatever.
Sure.
We have it all, the formats are there and the repositories are there and whatever. It wouldn't be a big problem, but it's a little bit of extra effort and authors aren't doing it. Publishers aren't willing to make their authors do it.
No.
And
I'm wondering.
I discussed. Sorry.
Sorry, I mustn't interrupt, but what? I was just thinking whether the fair thing is going to bring some more pressure on that side.
Yes, true, in a way. Yeah, I mean, essentially it's a requirement. Perhaps the pressure comes from funders or so, I don't know.
Yes, very much so. That's almost the only pressure that works.
Do you think that's better than the publisher pressure? Because I mean, the publisher one is, I mean, otherwise you cannot publish, right? The funder one, I mean, does it really get followed up afterwards? Do the funders check that?
Well, no, I mean, clearly the Wellcome Trust not going to dig down into NMR outputs from instruments. So I don't know who's going to do that, or even smiles from the chemists that they're funding. So I don't know.
But
who knows,
I mean,
for the future?
If it comes to the checking, in a way that should be automated. So it shouldn't be a problem to automatically check if certain
Absolutely right. Yeah, absolutely right. So you could, if you could get hold of the Word version or the Word version of the manuscripts and CSV files, yes, the auto checking and auto markup would be, we know it could be doable, but which publishers are going to move with this?
Yeah.
Well, I think we should sidestep the publishers because they've been asked to do this for 20 years and there's no sign of them doing it.
Yeah.
The pressure comes from, first of all, the pressure for open, which is having an effect.
Yes. Yeah.
And Europe in particular is strong on this and requiring people to be open. And some of the countries, Germany in particular, is putting money into requiring major outputs to be published as open. Secondly, I think there are large untapped areas, particularly theses. So much of this is...
What was that, Peter?
In theses.
Oh,
graduate
sorry.
theses.
Yeah, fine. Yes, absolutely.
And so often the material is collected in a thesis before it is published in the literature, and it's actually fairly straightforward and usually better to get it from a thesis than it is from a published document.
Yeah.
And so that's an area. We've ran a project 2 months ago. This was from the British Library, where we looked at 100,000 UK theses and extracted all of those into an indexing system. So it's possible to do it. And the same is true in other grey literature material. So a lot of stuff around which is attractive. And what you've got to do is find somebody who desperately needs to do it and is prepared to put some effort in. It's not going to happen by wishing other people to do it. It's going to happen by enthusiasts continuing to push and so it's possible.
I mean, regard to theses, it's just what's his name? He's one of my contacts at on Twitter, but he's just extracted the thesis, all the chemistry from the thesis in the University of Alabama. And he's now put them into Pubchem. So that's a step
exactly
in the direction. I suppose sucking it out of ELNs is short-circuiting publishing altogether in some senses, but that raises its also technical issues, doesn't it, of the veracity of pulling stuff out of ELNs.
Yeah. Okay. So thanks a lot for the discussion, everyone. I would suggest if you are interested in those areas, feel free to get in touch with Chris or Peter. I've just seen Peter was posting his email address as well. So feel free to get in touch with them. So I would like to thank both of the speakers, Morgan and Chris, much appreciated. Thanks a lot for your time. And our next meeting will be on the 25th of November. So we will have one speaker is defined already, Zachary Del Rosario. It's about model validation. So you don't want to rediscover what you know already, but you want to discover something new. So how do you evaluate a model actually to take that into account? So that's one speaker, but there's still one more speaker slot left as well. So if you would like to present, let me know either at this meeting or at future meetings. So thanks a lot, everyone. Please do stay on the line because we will now have our virtual pub visit, so to speak. So I will put people into breakout rooms. And so please stay on the line, see who you meet. It might be some of the speakers, it might be people you don't know yet. And so please do stay on the line. Okay, thanks a lot, everyone, for joining. Thank you. Talk to you soon.